Hello, Namaskar, and very good afternoon to all the viewers connected with us on NCERT's live and interactive program. I am Anu Gupta, your host for this session. Dear viewers, हम इस 1 घंटे के लिए आपसे जुड़ गए हैं क्लास 11th के सब्जेक्ट बायोलॉजी के टॉपिक द लिविंग वर्ल्ड एंड बायोलॉजिकल क्लासिफिकेशंस के लिए. I would like to tell you that we are streaming live on PMA Vidya channel number 11. Apart from that, if you wish to catch us online, you can also go to our YouTube channel which goes by the name NCERT Official. You can become a part of our live interactions by posting in your queries, feedback and questions in the live chat box of our YouTube channel. So, जैसा कि दर्शकों मैंने आपको बताया इस टॉपिक के बारे में लिविंग वर्ल्ड और बायोलॉजिकल क्लासिफिकेशन. When I think about it, dear learners, our world, our universe is filled with variety of species of living organism, be it microorganism, be it the largest mammals. तो इन्हीं सब बातों को डिटेल में बताने के लिए हमारे साथ स्टूडियो में एक्सपर्ट जुड़ गए हैं. आइए मैं उनसे आपको मिलवाती हूँ. हमारे साथ आज जुड़े हैं डॉक्टर पुनीत शर्मा. सर इज असिस्टेंट प्रोफेसर डिवीजन ऑफ एजुकेशनल किड्स NCERT न्यू दिल्ली. सर नमस्कार, आपका बहुत बहुत स्वागत है हमारे स्टूडियो में जी. साथ ही मैं वेलकम करना चाहूँगी हमारे इंडियन साइन लैंग्वेज एक्सपर्ट्स जो हमारे साथ जुड़ी हैं. हमारे साथ आज जुड़ी हैं नैना और मिस अंबू साक्षी. अभी आप नैना जी को अपने सामने देख रहे हैं स्क्रीन पर. बहुत बहुत स्वागत है नैना जी आपका. Dear viewers, as I already told you the topic, so if you wish to connect with us telephonically also, do write down our mobile number which is flashing on the screen right now. It is 8800440559. Also, if you wish to write to us in detail, do note down our email address which is class specific. It is dth.class11@ciet.nic.in. So दर्शकों और मेरे प्यारे बच्चों, बिना किसी देर के आइए चलते हैं डॉक्टर पुनीत के पास जो कि आपको बायोलॉजी में लाइफ, लिविंग वर्ल्ड और बायोलॉजिकल क्लासिफिकेशन के बारे में और जानकारी देंगे. सर
नमस्कार मैम, थैंक यू सो मच. I would like to know about today's topic, the living world and biological classification. So these are two topics of grade 11, chapter 1 and chapter 2. So together we will be trying to understand about these topics and their concepts related to biology, the living world and the classification of various living organisms and about their concepts.
राइट सर, थैंक यू सो मच बताने के लिए हमारे दर्शकों को कि आज हम क्या क्या टॉपिक कवर करने वाले हैं. क्या हम शुरुआत कर सकते हैं बायोलॉजी से कि हम बायोलॉजी से क्या समझते हैं?
जी ज़रूर. बायोलॉजी इज़ अ वर्ड मेड अप ऑफ 2 words. बायो मीन्स लाइफ एंड लॉजी मीन्स टू स्टडी. इसको हम हिंदी में जीव विज्ञान कहते हैं. जीवों के बारे में पढ़ना वो विज्ञान जीव विज्ञान कहलाता है. The study about life, living organisms and living structures, it is called as biology.
Right. तो सर आगे बढ़ते हैं सेशन की तरफ.
जी.
Now
I would like to concentrate on, already we have discussed about the biology. So today's focus will be the living world and its biological classification. So in relation to this, first of all we would like to understand about life. What is life? And to whom we will be able to call living. जीव और जीवन क्या है उसके बारे में, उसके गुणों के बारे में थोड़ा सा हम जान लेते हैं. सबसे पहले जो भी लाइफ है उसके कुछ पर्टिकुलर कैरेक्टरिस्टिक्स हैं. फॉर एग्जांपल ऑर्गेनाइजेशन, मेटाबॉलिज्म, होमियोस्टेसिस, ग्रोथ. दीज आर सर्टेन प्राइम कैरेक्टरिस्टिक्स which are again having certain different other characters also: adaptation, response to stimuli, reproduction. Now I would like to explain about organization. Organization की जब हम बात करें तो यहाँ पे organization एटम मॉलिक्यूल से स्टार्ट होके हमारा जो बायोस्फीयर है वहां तक जाता है. ये अपने आप में एक हायरार्की है, बहुत छोटे से एटम मॉलिक्यूल से लेकर और हमारा जो बायोस्फीयर है, पूरा जो जीव मंडल है, पूरा जो अर्थ है, अर्थ के चारों ओर जितना भी है वो सारा बायोस्फीयर कहलाता है. तो एक छोटे एटम से लेके जब हम आगे बढ़ते हैं हायरार्की में स्टेप बाय स्टेप, तो एटम्स मॉलिक्यूल्स बनाते हैं आगे चल के कुछ लिविंग स्ट्रक्चर्स को, कुछ बायोमॉलिक्यूल्स को. वो बायोमॉलिक्यूल्स फर्दर कुछ ऑर्गेनल्स को, कुछ पार्ट्स को बनाते हैं। वो ऑर्गेनल्स जो सूक्ष्म, जो छोटे, जो माइक्रोस्कोपिक होते हैं, वो फर्दर एक स्ट्रक्चर को बनाते हैं जिसे हम यूनिट ऑफ लाइफ, स्ट्रक्चरल एंड फंक्शनल यूनिट ऑफ लाइफ सेल कहते हैं। तो ऐसे सेल एग्रीगेट होकर टिश्यू एंड फर्दर सो एंड सो। सो अकॉर्डिंगली दीज आर द कैरेक्टरिस्टिक्स ऑफ लाइफ, विच इज फर्स्ट वन इज द ऑर्गेनाइजेशन, सेकंड इज मेटाबॉलिज्म। नाउ व्हेन वी हैव ऑल दीज स्ट्रक्चर्स as a cell, now certain work has to be done to produce energy. So all of those chemical reactions, biochemical reactions particularly, which are happening in the living organism, they constitute the metabolism.
Right.
And metabolism consists of two types of metabolism. One is anabolism and another is catabolism. So anabolism is related to creation, so creative biochemical reactions, and anabolism is related to catabolism is related to destructive reactions which break down the certain biochemicals.
Right.
Next is homeostasis. So after all these things, certain environment is there for all living organisms. सभी जीवों के चारों ओर कुछ ना कुछ एक वातावरण है। अब उस वातावरण में चेंजेस, परिवर्तन आते जाते हैं। So there are changes happening in the environment. So the organism's body is supposed to keep a balance of all these metabolism as required. So this balancing of all biochemical reactions, the internal environment which is not adversely affected by the outside environment, is called as homeostasis. So this equilibrium is required. And next is when after all these things happen, we move towards the increase in the mass, increase in the cell, टिश्यू विच accompanies द ग्रोथ, जो ग्रोथ से संबंधित है, वृद्धि से संबंधित है। आगे चलते हैं। जीव क्योंकि एक जगह पे रहता है, लिविंग इन अ पर्टिकुलर एरिया, अडेप्टेशंस आर रिक्वायर्ड एक्सटर्नल एनवायरनमेंट के अकॉर्डिंगली, तो उसमें कुछ ना कुछ ऐसे ट्रेट्स, कैरेक्टर्स होते हैं। रेस्पॉन्स टू स्टिमुलाई, उसको कहीं ना कहीं जो कुछ ना कुछ उसके बिहेवियर चेंजेस में कुछ ना कुछ जो फोर्सेस उसको मिले, उसके अगेंस्ट में या उसके पैरेलल में उसको रेस्पॉन्स करना होता है। एंड फर्दर अपनी एंटिटी को कंटिन्यू रखने के लिए नई संतति, नई ऑफस्प्रिंग को प्रोड्यूस करना होता है, जो कि पार्ट है रिप्रोडक्शन का। आगे बढ़ते हैं। क्या हम इन सारे कैरेक्टरिस्टिक्स में से हमारे डेली लाइफ में कौन से ऑब्जर्व करते हैं, ये बच्चों के लिए क्वेश्चन है। नेक्स्ट, मूविंग टू द स्लाइड इज characteristics of life, already what we have discussed. Now this question arises in the mind of a student, whether is a virus alive or not. So virus is that entity which is a connecting link between the living and non-living. So whenever it is present in a host, it retains as a parasite, may be internal or external, and then it is live and it completes all the metabolism and replication inside that host body. But whenever it is outside the host body, it is alive, it is a non-living entity. So why it is a connecting link between the living and non-living. Next is when we discuss about the characteristics of life, there are so many different organisms, plants, animals, microorganisms we are finding surrounded by the environment. So basically to understand this, so to understand this, we would like to go to biodiversity. What is biodiversity? Fine. Basically? So bio means life again, and diversity, that is variations. We found variations at different levels, at genetic level, at species level, at ecological level also. So all these differences, they constitute the biodiversity. So in simple words, if we would like to say, it is variety of living organisms in our surroundings. And the number of species that are existing in the world today, right now what we have explored, it is around 1.7 to 1.8 million, which is only 10% we have discovered. Next is,
that's a very big figure, I must say.
It's a very big figure. Only 10% yet we have discovered.
सर, अभी आपने वायरसेस की बात की, तो जैसे हम कोरोना वायरस को भी जानते हैं, हम सभी जानते हैं और जी कर आए हैं उस समय को जहाँ पर कोरोना वायरस हमारे बीच में था, तो कुछ उसके बारे में भी सर कनेक्ट कर बताइए।
जी, कोरोना वायरस इज अ सिंपल, अ कॉमन कोल्ड वायरस, नंबर वन. Nothing to fear about coronavirus. It is a kind of a strain or a type of a common cold virus. So whatever precautions we are taking in general for the common cold, we are isolating ourselves, we are protecting ourselves from the mask, and so we
Washing
are
hands.
supposed to washing hands.
Ji.
So since a common cold is also infectious disease,
Right.
so when it is not in control, it can cause pneumonia also.
Right.
So the same way we are supposed to take care regarding corona also. So coronavirus is a simple, nothing to fear about. It's a common cold strain.
Right.
Yeah. So moving to again biodiversity part. So India is also a rich biodiverse country, and the richness is shown in the ways of hotspots. So India is also having certain hotspots. What are hotspots? Hotspots are those areas which are having high richness of species of organisms plus endemism, that is these species are found only in that particular area.
Right.
Next, we are having these, there are 4 types of biodiversity hotspots in our country. One is the Himalayas, second is the Western Ghats, Indo-Burma and Sundaland, which constitute of the Andaman and Nicobar area, which can be depicted through this map also.
Right.
And there are total 17 mega diverse countries in world, and India is also one of the mega diverse country. Taxonomy. Now, since we are moving towards, we have studied about, we have gone through the life, living characteristics, and then biodiversity. So there are different organisms found on the earth. Now, how we will going to study about them? 1.7 million to 1.8 million. To study them properly, scientifically, to know about them, to know about their importance, we are supposed to do certain scientific process for these, so which is called as taxonomy.
Right.
Taxonomy constitutes of 3 parts, which is identification, classification and nomenclature. So identification means to identify which particular species it is, basically an animal or a plant or a microorganism. So to identify, it is identification. Next is nomenclature. On the basis of certain principles to name these species, it is called as nomenclature. And finally, to put them under a certain group,
Right.
where they will exist, where they will lie in that particular taxonomy, it is called as classification. So these are the 3 parts of taxonomy, that is identification, classification and nomenclature. So now identification, how we will be able to identify the living organism. So there are certain methods and ways. One I have shown on the screen, it is the keys. So there are certain statements which are represented as keys, and they are generally present in the form of couplet, means contrasting characters,
Ji.
whether this character is found or not found. So
Right.
this is a contrasting character. So if it is present, then we will move to the next key, which is asking again a contrasting statement, and where it is found, and so we will be moving to the keys, and finally delimiting ourselves and narrowing the identification to the species level. This way we will find out which type of organism it is basically. While moving through this taxonomic keys, these are the ways we can utilize the keys. And classification. Now, when we have find out the kind of organism, what type of organism it is basically, so classification कैसे करें, कैसे हम उसको classify करें, उसके क्या आधार हैं, क्या basis हैं. So basis, 2 important bases are there, that is similarity and dissimilarities in the characteristics, whether these characters are similar in between 2 organisms or not, this is dissimilarity again. And then evolutionary relationship, उनके एंसेस्टर्स कौन से रहे, कहाँ से चल कर आए वो भी हमें देखना होगा, एंड देन रिलेटेड ग्रुप्स में उनको हम रखेंगे, एंड दिस इज बेसिस ऑफ द क्लासिफिकेशन, एंड विच कॉन्स्टिट्यूट ऑफ द सिमिलरिटी, डिसमिलरिटी एंड एवोल्यूशन. नेक्स्ट मूविंग टू द नॉमेनक्लेचर पार्ट, that is naming, scientifically naming the living organism, it is called as nomenclature, and again it is based on certain rules. Carolus Linnaeus was the first scientist who has devised the binomial nomenclature binomial. Bi means 2. जिस scientific name में जहाँ पे 2 शब्दों का इस्तेमाल होता हो, 2 words are used. The first word which is representing the genus and the second word which is representing the species. So this is generic name and specific name, which is also called as specific epithet. epithet is that word which let the student to understand about the characteristic of the genus.
Right.
It tries in that way. This name is particularly given in such a way which shows certain characteristics of that particular genus. So why it is called as specific epithet. I have proposed a certain example also, given example for binomial nomenclature of mango, that is Mangifera indica. So Mangifera word is the genus name,
Right.
and indica is the species name or the specific epithet that it may be find out at certain place,
Found, yes,
certain
yes.
characters. So it is representing that particular characteristic.
All right.
So international principles are there for and criteria are also there for nomenclature, and it has been governed by ICBN, International Code for Botanical Nomenclature, and International Code for Zoological Nomenclature for the naming of the plants and for the naming of the animals respectively. So these are the certain rules and regulations which are given in ICBN and ICZN codes.
Right. So sir, a question comes to my mind.
Yeah.
आपने भी ये भी दिखाया था कि जैसे मैंगो का एपिथेट इंडिका है,
जी।
सो क्या मैंगो ऑल अक्रॉस द वर्ल्ड इसी एपिथेट से जाना जाता है?
नहीं।
Okay.
क्योंकि मैंगो की बहुत सारी जाति या प्रजातियां होंगी, स्पीसीज होंगी,
जी।
तो उनके अपने अपने अलग अलग नाम होंगे।
Alright.
मैं इसका एक आपको एग्जांपल देना चाहूँगा। तेंदुआ, लेपर्ड, पैंथर जिसको हम जानते हैं, तो पैंथर जो है उसका जो जीनस है वो पैंथरा है।
राइट।
तो पैंथरा, उसके बाद में जो उसका स्पेसिफिक एपिथेट है वो पार्डस है।
ओके।
अब उसके बाद में भी ये पाया गया कि पार्डस के बाद में भी ये पाया गया कि कहीं ना कहीं देयर आर डिफरेंसेस इन द स्पीसीज दैट गोज टू सब स्पीसीज लेवल। सो व्हाइल गोइंग studying about the panther in India and Sri Lanka, it has been found that Indian panther, they are called as Panthera pardus fusca. So fusca is that particular subspecies which is representing the pardus species.
Right, right.
So when we are having this species, it means it shows that it is having certain different traits or characters, but it is coming in the same genus.
जी।
Yeah. So universal rules of nomenclature are also prescribed and given, and it says that each binomial or the scientific name, it has to be Latinized. The language used in science, scientific terms, it is Latin, and if name is not in Latin, it has to be Latinized, and two words should be there, one representing the genus, another representing the species. And these, when these two names, the scientific name or the biological name, when they are written, they are separately underlined. It is again a rule given in the universal rules of nomenclature, and when it is printed, it should be in italics. It is again compulsory to write these names in such a way only. And while writing the name of the genus, the first letter has to be always in capital and rest of all in small. And while writing the name of the species, all letters will be in small letters only. जब भी हम जीनस का नाम लिखेंगे तो पहला अक्षर हमेशा कैपिटल होगा, और जब भी हम स्पीसीज का नाम लिखेंगे तो सारे ही लेटर उसके जो है स्मॉल होंगे. सो एक्सेप्ट ओनली द फर्स्ट लेटर ऑफ जीनस, ऑल लेटर्स विल बी इन स्मॉल. स्पेसिफिक एपिथेट जो हमने बात करी जैसे मैंजिफेरा इंडिका के लिए, तो इंडिका यहाँ पे उसका स्पेसिफिक एपिथेट को रिप्रेजेंट करता है. नाउ हमने नॉमेनक्लेचर कर लिया, अब हम क्लासिफिकेशन की तरफ अगर बढ़ते हैं तो हम कैसे उनको ग्रुप्स में रखेंगे, कहाँ उनको किस ग्रुप में रखें, ये हम आइडेंटिफिकेशन के आधार पे पता लगाएंगे. विल फाइंड आउट द वेज ऑफ डिफरेंट वेज ऑफ आइडेंटिफिकेशन टू इन विच ग्रुप वी कैन कीप दीज लिविंग ऑर्गेनिजम्स. सो while putting them in certain groups, coming from top to bottom, these are called as labels. And they are in, since they are in hierarchy, they are in from top to bottom or maybe from lower end to top end. So this constitutes the hierarchy, and this is the hierarchy of taxonomy. This is the, so why it is called as hierarchical taxonomy, taxonomic hierarchy. And while coming from top to bottom, the topmost hierarchy or the level, which is also called as rank and which is also called as taxon. इनके और भी नाम दिए हैं इस लेवल के, इस पद के, इस जगह के, इस स्तर के, और इसको कहते हैं टैक्सॉन भी कहा जाता है, लेवल भी कहा जाता है या रैंक भी कहा जाता है. टैक्सॉन एक कॉमन वर्ड यूज होता है जो कि कॉम्पिटिटिव एग्जाम में पूछा भी जाता है. नाउ द हाईएस्ट टैक्सॉन, हाईएस्ट लेवल इज द डोमेन, and domain is that level which constitutes all of the life of biosphere. And when we are coming next to domain, it is having certain kingdoms, like for example plant kingdom, animal kingdom. So there are certain different kingdoms, and then these kingdoms consist of various phylum in the case of animals, and in the case of plants the name in case of this phylum it is division. So for plants in replace of phylum we are using word division. And these phylum and division consist of so many classes. So we are moving from top to down and coming to the smallest level, smallest taxon or the rank, which is called as species. So this species, the smallest level of any living organisms while doing this hierarchical taxonomy, and this is the only natural taxon in this taxonomic hierarchy. Rest of all domain, from starting from domain till genus, they are all artificial. As such, nothing is found in nature in such a way. We have created for our convenience, for scientific study, for identification, but species we found in nature naturally. different species. हम बोलते भी हैं कि देखो ये अलग सा दिख रहा है जाति, ये डियर की अलग जाति दिख रही है, तो ये naturally पाई जाती है. सो ये कॉम्पिटिटिव एग्जाम का क्वेश्चन भी है, which is the natural smallest sub unit of classification. It is the species. So taxon rank level we have understood. Taxonomic hierarchy we have already discussed, which consists of different while starting coming from the top to bottom, they consist of different groups, and while coming from top to bottom the relatedness is increasing. When we have started from the top, all were life. They were not similar in such a way, they were life, living, so they were similar in that way. While coming to the next level, that is kingdom, now we found no, this is plant, this is animal. So they are having different characters. On that basis we are saying that okay, this is plant, this is animal. So in such a way we are moving downstream and reaching to the smallest level of this classification, which is a species. It means that when we are discussing a species, they are similar in so many ways. Even at genetic level also they might be having a much of similarity. So this is called as a species level. Next is taxonomic aids. Now when we are doing taxonomy, we require certain help, certain assistance, which are called as taxonomic aids. And there are certain examples, like for example herbarium, botanical gardens and museums. So these are those ways, those aids which help out in doing taxonomy also and preserving these specimens also for further future records. So first is the role of herbaria in taxonomy. So herbaria is basically a collection of dried and pressed plant. It is a collection of that all these dried and pressed plants, and it is done on herbarium sheet, which is made up of a good quality paper, acid free paper. It is almost 0.7 to 0.9 mm of thickness, and it is in general standard size is around 30 centimeter to 40 centimeter, the size of the herbarium sheet, on which we have this embossed this dried and pressed plant, labeling the features and characteristics of that plant, and who has identified, who has collected this plant, and all such entries we are supposed to put on that label. Moving to the botanical gardens. Botanical gardens is again a collection of different plants which are utilized in taxonomy. So it has been preserved in botanical gardens also. So further studies can be done, and further whosoever wants to study okay what characteristics they are representing, so they can go those botanical gardens and study about these plants also.
सर जब आप हर्बेरियम की बात कर रहे थे तो मुझे अपना बचपन याद आ रहा था. कई बार हमें एक्टिविटी इस तरह की साइंस में दी जाती थी जहाँ पर सर्टेन टाइप के प्लांट्स को पेपर में पेस्ट कर उसके ऊपर आई थिंक फिल्म चढ़ाकर लेकर आने को कहा जाता. तो क्या ये वही चीज है हर्बेरियम?
सही, बिल्कुल. हमें देखिए, हमारी जो एजुकेशन रहा, हमारी क्लासेस में प्रैक्टिकली बहुत सारा हमारे टीचर्स ने अच्छा काम करा है. हम जब छोटे थे, जब इतना हम डिटेल्स नहीं जानते थे, लेकिन हमें हमारे टीचर्स ने कुछ प्लांट के पार्ट्स को किताब के बीच में रख के ड्राई करके प्रेस करके रखने के लिए कहा, ताकि हम उसको फिर एक कलेक्शन के रूप में एक शीट्स में लगा सकें और हम बता सकें कि ये लीफ किसकी है, इस लीफ में में इसकी क्या कैरेक्टरिस्टिक्स हैं। तो इसी तरह से प्लांट और प्लांट के पार्ट्स को यहाँ हर्बेरियम शीट के ऊपर लगाया जाता है। डिफरेंस इतना ही है बस कि यहाँ जो शीट है वो स्पेसिफिकली टेक्सोनॉमी के काम के लिए ली जाती है, उसकी अपनी एक क्वालिटी होती है।
राइट
जी। So moving to the recording the plant descriptions also, we are having different types of records: flora, manuals, monographs, catalogs. So flora is particularly that record which is for a particular area. In this particular area, these are the plant species which are found in this area having certain characteristics. It is called as flora. While manuals, they are giving a general idea about the plants, how the plants, different types of plants. So to identify which type of plant it is found in my area, we will utilize the manuals. And monographs are dedicated for specifically a taxon, a level or a rank. Let's see, if I'll discuss genus, so I'll discuss one genus only in particular monograph. So this is monograph. And catalogs are ready reckoners, pamphlets or leaflets which gives a record description of those plants, and to identify immediately at the spot only, these are the catalogs. Methods of, since we have discussed about the plant, and in the same way animals are also preserved for further taxonomy, and so they have been preserved in various ways. Number one, which is very common to preserve the specimens of the animals on the basis of which we can study the taxonomy, the characteristics, is preserving them in solution. And very common solution is formalin, which is aldehyde, HCHO. So a percentage of formaldehyde is utilized. It is dissolved in water to make it dilute solution in a proper way. It is having a standard percentage to mix formaldehyde, so as it can preserve the tissues of the organism. So the tissue will not decomposed, the protein will be intact for that particular tissue, and they will not be damaged in coming time. So we can see this specimen as it is protected in that particular formalin. Next is we have given certain examples in a tabular form for this taxonomic hierarchy, having certain classification also. For example, man, Homo sapiens, it is the biological name of man, and which comes under the genus Homo, and we represent to a family Hominidae, and order Primata, class Mammalia, phylum division in the case of plants, and phylum in the case of animals. So phylum is Chordata. So these are having certain characteristics, then only they have been grouped in such phylum or such classes.
Right.
Moving to the biological classification, why there is a need of biological classification, how it will be helping us, and why we are doing biological classification. Already we have gone through the classification. So there are certain features or characteristics due to which we are doing this biological classification. First of all, to understand about the evolutionary relationship. Next is to find out the identification parameters of that particular organism, and how to study simply those organisms. It is required to classify accordingly, to understand their ecological roles also, and to aid in medical and agricultural fields also. We are doing this classification of all living organisms. So biological classification, when we will discuss how it has been started. So Aristotle, he was the first scientist who has started classifying the living organisms, and he has classified both plants and animals separately. So on a very raw basis, Aristotle has classified plants on the basis of their morphological characters. Morphological means which we are able to see through eyes, जिनको हम बाहर से देख पा रहे हैं, वो जो है वो मॉर्फोलॉजिकल कैरेक्टर्स कहलाते हैं. तो मॉर्फोलॉजिकल कैरेक्टर्स के आधार पे ऑन दिस बेसिस एरिस्टोटल हैज क्लासिफाइड प्लांट्स इनटू 3 ट्री, श्रब्स एंड हर्ब्स, जो उनके अपने अपने कुछ डिफाइंड कैरेक्टरिस्टिक्स हैं. ट्रीज बहुत टॉल होते हैं, श्रब्स के, हर्ब्स के अपने कुछ कैरेक्टरिस्टिक्स हैं. एंड द सिमिलर वे एरिस्टोटल हैज डन क्लासिफिकेशन फॉर एनिमल्स, एंड he has classified animal on the basis of presence of red blood and absence of red blood. If it is present, it is called as enema, and when it is absent, it is called as anemia. And so why we are having this term anemia.
Okay.
That is absence of
Right,
red blood. So
right.
anemia. So Aristotle has started using this word. Next comes, since it was not exactly right way of classification, more improvement was required. So two kingdom classification was given by Carolus Linnaeus, and he has classified simply mere in plants and animals. Again, other organisms were left behind. So to classify further, to include further remaining leftover organisms, living organisms, three kingdom classification was given by Ernest Haeckel, four kingdom classification was given by Herbert F. Copeland. When we are talking about three kingdom classification, so plant, animal, and then Ernest Haeckel has included Protista also, Monera also. And when we discuss about four kingdom classification, Herbert F. Copeland included plant, animal, Monera and Protista. We will be coming on these terms also later on in our coming slides. And finally, the most acceptable and followed classification right now which we are following today, it is five kingdom classification, which was given by R. H. Whittaker in year 1969. Now moving to this five kingdom classification. So what were the basis of classifying organisms? So you can see the characters. There are certain 5 to 6 characters we have given in the table. On that basis we are classifying the living organisms into 5 kingdoms: Monera, Protista, Fungi, Plantae, Animalia. So first character is the cell type. There are two types of cell on certain basis. When we are saying types of cell, we can classify cell or types of cell, we can say in so many ways. But while having this classification, we can understand that okay, there are two certain types of cells which are called as prokaryotic cell and eukaryotic cell. प्रोकैरियोटिक सेल, प्रोकैरियोट शब्द से बना प्रोकैरियोटिक सेल. प्रोकैरियोट, प्रो मीन्स प्रिमिटिव. प्रो जब भी प्रीफिक्स आएगा किसी के लिए, इसका मतलब होता है प्रिमिटिव, जो आदि काल में, जो बहुत शुरुआत के समय में था, उस तरह का, दैट इज कॉल्ड एज प्रो. एंड कैरियोटिक में कैरियोट शब्द निकला है कैरियन से. कैरियन मीन्स न्यूक्लियस. तो वो सेल जिनमें न्यूक्लियस प्रिमिटिव टाइप का था. नाउ क्वेश्चन कम्स, प्रिमिटिव टाइप का कैसा होता है? प्रिमिटिव टाइप मतलब जिसके चारों ओर न्यूक्लियर मेम्ब्रेन एब्सेंट है, जिसके फर्दर जो अदर सेल ऑर्गेनल्स हैं उनके चारों ओर भी न्यूक्लियर मेम्ब्रेन जो है नहीं पाई जाती है, न्यूक्लियस के चारों ओर न्यूक्लियर मेम्ब्रेन नहीं है, और इस तरह के जो न्यूक्लियस हैं वो प्रोकैरियोटिक न्यूक्लियस कहलाते हैं, और इस तरह के जो सेल है वो प्रोकैरियोटिक सेल कहलाते हैं. तो जो ऑर्गेनिजम्स इस तरह के सेल से बने होते हैं उन्हें प्रोकैरियोटिक ऑर्गेनिजम्स कहते हैं. सो मोनेरा प्रोकैरियोटिक ऑर्गेनिजम्स हैं क्योंकि इनमें इस तरह का सेल पाया जाता है. फर्दर मूविंग टू प्रोटिस्टा, फंजाई, प्लांटा, एनिमिलिया, दे आर हैविंग यूकैरियोटिक टाइप ऑफ सेल. EU is a prefix which says true, means आज की तारीख में, प्रेजेंट में किस तरह का न्यूक्लियस पाया जाता है, कैसा पाया जाता है, जिसके चारों ओर वेल डेवलप्ड न्यूक्लियर मेम्ब्रेन पाई जाती है, जिस सेल के अदर सेल ऑर्गेनल्स जो हैं वेल डेवलप्ड हैं, जिनके चारों ओर उनकी अपनी अपनी मेम्ब्रेन्स हैं और बहुत अच्छे इवॉल्व हो चुके हैं. इस तरह के जो सेल्स होते हैं वो यूकैरियोटिक सेल्स कहलाते हैं. और ये हमारे रेस्ट ऑफ जितने भी 4 किंगडम्स हैं, दैट इज प्रोटिस्टा, फंजाई, प्लांटे, एनिमिलिया, इन सभी में यूकैरियोटिक सेल पाए जाते हैं. नाउ सेकेंड क्वेश्चन कम्स, सेकेंड कैरेक्टर कम्स अबाउट द सेल वॉल, कि ये सेल है, इसके चारों ओर सेल वॉल है या नहीं है, और है तो किस केमिकल की, किस बायोकेमिकल की बनी है, ये भी डिफाइन करता है किसी एक पर्टिकुलर ऑर्गेनिज्म को किस किंगडम में हम रखें, ये डिफाइन करेगा. सो वी कैन सी इन द एग्जांपल फॉर द मोनेरा, नॉन सेल्यूलोजिक सेल वॉल पाई जाती है. इट मीन्स सेल वॉल इज प्रेजेंट बट इट इज नॉन सेल्यूलोजिक, मीन्स सेल्यूलोज की नहीं बनी है. हम जनरली प्लांट्स के लिए पढ़ते हैं हमारी छोटी क्लासेस में कि प्लांट सेल के चारों ओर सेल वॉल पाई जाती है जो सेल्यूलोज की बनी होती है. सो इन द केस ऑफ मोनेरा, the organisms which belongs to the Monera, the cell wall is not made up of cellulose. It is made up of polysaccharide and certain amino acids. In the case of Protista, it is present, but certain now it is different from others. In the case of fungi, the cell wall is made up of chitin, and in the case of plantae it is a cellulosic cell wall, but in the case of animal, the animal cells are devoid of cell wall. Cell wall is absent in the case of animal cell. It is again a characteristic. So wherever we will be finding this absence of cell wall, we will definitely say, okay, this might be an animal.
Right.
The same, the third, in the same way, the third characteristic is the nuclear membrane. So nuclear membrane, as we have already discussed, in the case of prokaryote it is absent, and in the rest of all eukaryotic cells it is present. Body organization, we can see certain organisms, they are very simple, only made up of one cell, single cell, or maybe a very simple cell. So prokaryotic single cell, it is only having a level of cell, so why it is called as cellular. So Monera consists of prokaryotic cellular level, and in the case of Protista, they are also made up of cell, but the cell is of eukaryotic type. That is true in nature in terms of well developed nucleus, in terms of well developed cell organelles. In the case of fungi, Plantae, Animalia, we are having multicellular level, means so many different types of cells are found which are further forming tissues, tissues are further forming organs, organs are further forming organ system, and finally the body. So this is the level of organization, body organization, while when we are discussing the 5 kingdom classification for Monera, Protista, fungi, Plantae and Animalia. In the case of the last, which is important characteristic while devising this 5 kingdom classification, it is mode of nutrition. What type of mode of nutrition of that particular organism is this? Is it autotrophic? Auto means self, trophic means the generation of energy or the food. So if an organism is generating its own food, it is called as autotroph. So it is autotrophic mode of nutrition. But an organism, if an organism it is not generating its own food, it is relying on certain other organisms, then it is relying on other. Hetero means other. So heterotrophic means for food or energy sources, it is depending on other organisms. So why it is called as heterotrophic mode of nutrition. Do you have any query regarding
नो सर, मैं रिकॉल कर रही हूँ, हमने ऑटोट्रॉप्स और हीटरोट्रॉप्स पढ़े हैं, आई थिंक 8th और 9th स्टैंडर्ड में ये टर्म्स से हमारे बच्चे फेमिलियर होंगे जरूर.
एग्री, यस. सो मूविंग टू फर्दर द डिस्क्रिप्शन ऑफ द मोनेरा. Monera usually consists of bacteria, and bacteria is having certain type of nutrition. On that basis, the bacteria can be further classified as autotrophic bacteria, chemosynthetic bacteria or heterotrophic bacteria. Already we have discussed heterotrophic means depending on other organism for their food or energy. So the majority of bacteria, they are heterotrophic bacteria, but certain bacteria, they are autotrophs also, like cyanobacteria, blue grain algae, and certain bacteria, they are chemosynthetic, means they are generating their energy through chemicals, not through light source. Since autotrophic, usually they are generating the source of energy for them is photo, that is light. So why they are called as photoautotrophic. Moving to the next is on the basis of shape of bacteria, they have been further classified as cocci, that is spherical in shape, vibrio, that is comma shaped, bacilli, rod shaped bacteria, spirilla, that is spiral in shape. So these are certain ways the bacteria has been classified on the basis of shape. Moving to the type of bacteria, there are bacteria which are called as archaebacteria. Archae word represent very primitive, ancient, which has started at the earliest. It is ancient.
So archaeology,
archaeology, ancient, very ancient. So archaebacteria, these are the ancient bacteria which are having certain feature characteristics to sustain in their hard environments. For example, halophiles, they can sustain in extremely salty environments, and in the same way thermoacidophiles, they can sustain in strong acidic conditions and hot acidic conditions also. They are having certain features, they are having certain cell walls in them which protect these organisms from the external environment. Methanogens, these are those ancient bacteria which produces methane gas. and they are very common in case of the ruminant animals, the cattles. Then moving to the eubacteria, EU means already we have discussed, that is true. The present, the most common bacteria, eubacteria. So cyanobacteria, heterotrophic bacteria, chemosynthetic bacteria, all of these are eubacteria which are showing the features of present bacteria, and already we have discussed that heterotrophic bacteria are the majority of the bacteria, and they may be harmful or they may be beneficial also. Now there is one organism, mycoplasma. It is not having a cell wall in them, and they are the smallest living cell. In the case of living organism, they can survive in anaerobic conditions, means in the absence of oxygen. ऑक्सीजन नहीं है तो भी ये सर्वाइव करेंगे, और ये इनकी जो टेंडेंसी है, ये पैथोजेनिसिटी होती है इनमें, ये पैथोजन की तरह एक्ट करते हैं. दे कैन कॉज सर्टेन डिजीजेस आल्सो इन प्लांट्स एंड एनिमल्स. Moving to the Protista, this particular group consists of 5 subdivisions. Protista, which includes chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans, and all these groups, they are having certain defined definite characters with them. But in a general, the general characteristics of Protista, they are having flagella and cilia also. Flagella and cilia are those structures which are utilized and which are used for movement particularly. Flagella are in general less in number but long in shape. Cilia are in more in number but they are small in shape. They are hair like structures, cilia, and cilia usually move in a row fashion. Back and forth movement they show, but in the case of flagella they show whip like movement.
Right.
So this is the difference in between flagella and cilia, but they are present in Protista for movement. Eukaryotic structure, already we have discussed about the Protista, that is they are made up of eukaryotic cell, and this Protista links to plant, fungi and animalia kingdom also in certain evolutionary relationship. Reproduction, they usually do reproduction by both asexual and sexual. Asexual, which requires only one parent to produce new generation. Sexual, which requires 2 different parents generating 2 different gametes and then further fusion and then producing the new offspring through zygote. This is sexual reproduction, and usually the Protista, they belongs to aquatic habitat, means they live in water, they are found in water. So they are most common in aquatic conditions. Next is the chrysophytes. We have discussed about the chrysophytes. Chrysophytes and further diatoms, their characteristics are almost similar. So when we focus to this diatoms, their cell wall is unique. It is made up of certain embedding of silica. Moving to dinoflagellates, they are having certain characteristics. Habitat, if we will see for the dinoflagellates, they are mostly marine. Photosynthetic, yes, they are photosynthetic, that is they are doing photosynthesis, they are producing their own food. Moving to the euglenoids, euglena basically belongs to euglenoid, which is also a connecting link between in plants and animals.
Okay.
Euglenoids basically show photosynthesis during the availability of light, but in the case of dark, they show heterotrophic mode of nutrition. Next is slime molds, which is again the kind of Protista found, and they are having saprophytic mode of nutrition. Sap means juice. When they are dissolving, they are depending on certain organic materials, dead and decaying organic materials. So they feed on organic materials, utilizing them in the form of a sap or a juice. So why it is called as saprophytic mode of nutrition. Now moving to the last group of this Protista, it is protozoan. Now protozoan having certain characteristic also, and they are having certain evolutionary relationship with certain primitive animals also. Their lifestyle is, they are usually predators or parasites. They depend on other organisms for their food or energy. They may be external parasite, they may be internal parasite. Internal parasite means they are living inside the body. External parasite means they are outside the body. And the mode of nutrition, already since we have discussed, it is heterotrophy. They are depending on other organisms for their food. Protozoon has been, again, this protozoa is again classified into certain classes, their divisions. They are amoeboid, there are certain amoeboid protozoons, flagellated protozoons, ciliated protozoons and sporozoons representing their characteristics. Amoeboid means they are having pseudopodia for the movement, flagellated means they are having flagella with them, ciliated means they are having cilia with them for the movement, and sporozoons means they are producing spores which are infectious also, which are pathogenic also in certain cases. Moving to the third kingdom, which is kingdom fungi. They are having certain specific characteristic, that is, they are heterotrophic in nature depending on other for their food or energy. Morphological diversity, they are found in diverse nature, they are having diverse characteristics. Moving to the economic importance, yes, certain fungi has been utilized for producing some medicines also, they are having certain medicinal values also, but in certain cases they are harmful also. Ecological impact, yes, they impact ecosystem through their decomposition. Since fungi, they do this process decomposition, that is degrading the dead materials and producing again the nutrients to that particular environment. The fungal structure, we will discuss about the fungal structure. They are having septate hyphae. Hyphae are a filamentous structure, and when these hyphae aggregates, they constitute mycelium, that is network of hyphae, and these thread like structures, the fungal bodies, they help in movement and taking the nutrition also, and fungi is found in diverse environment in our surrounding. Fungi can obtain food through saprophytic mode of nutrition, through parasitic mode of nutrition, or through symbiotic mode of nutrition also. Now, here one new word has come, that is symbiont, or that is symbiotic mode of nutrition. Symbiotic, symbiosis means mutual cooperative aggregation. So, one example we will discuss in further coming slide for symbiotic association. When we move to fungal classification, they have been classified into 4 classes, subdivisions: phycomycetes, ascomycetes, basidiomycetes and deuteromycetes. So, they have certain general characteristics with them. For example, phycomycetes, they are primitive fungi with simple structures and aquatic habitats, and usually they are simple form of fungi. Ascomycetes, they are characterized by sac-like structure, and they usually form spores through which reproduction happens. Basidiomycetes, they are having basidiospores, and they are club-shaped fungi, and due to this structure, and they form spore also. Deuteromycetes, fungi lacking a known sexual reproductive cycle, since fungi reproduces by both sexual and asexual mode of reproduction. So, deuteromycetes are lacking somewhere known sexual reproductive cycles also. Moving to the what type of reproduction is occurring in the fungi. Both asexual and sexual mode of reproduction is occurring in fungi, and asexual mode of reproduction involves the spore formation, and there are certain different types of spores like conidiospores, sporangiospores, zoospores, and they are produced in certain bodies which are called as fruiting bodies. Sexual reproduction, it happens in the form of oospores, ascospores, basidiospores, and which depends on the type of class. Like ascomycetes, it is having ascospores in them, so why it is called as ascomycetes, since they are producing ascospores as a sexual structures for sexual reproduction. Moving to the kingdom Plantae, and kingdom Plantae is having certain defined general characteristics. First is alternation of generation, that is, a body might be having a haploid stage, a body might be having a diploid stage. Haploid means the condition which is represented by N, diploid means which is the condition represented by the 2N, and this condition is on the basis of the chromosome. As we know, the chromosome, it is the genetic material.
Right.
When it is paired, it is called as diploid, 2N. When it is unpaired, half, it is haploid. As we can see, it is in very common in the case of gametes, the reproductive cells, in our case the sperm and the ovum, they are haploid in nature. Since and rest of our all cells, they are diploid in nature, since they consist of paired chromosome. As we know that humans, we are having 46 chromosomes in
yes.
23 pairs. So when it is paired, it is called as diploid condition, and when it has been halved through meiosis division, through reduction division, it becomes 23 in the gametic cells or the reproductive cells. Then, and in this case it is called as haploid cell. So the same way, plants alternate between diploid and haploid conditions, generations. It is called as alternation of generation. Now, plant groups includes algae, that is Thallophyta, Bryophyta, Pteridophyte, and gymnosperms and angiosperms on the basis of defined characteristics. They are having certain heterotrophic members also, insectivorous plants also. In the case of plants, they do, yes, they are in majority, they are photosynthetic, they are autotrophic, they produce their own food, they do this photosynthesis, which is trapping the sunlight energy and converting it into chemical energy, photosynthesis in simple way, and they are having eukaryotic structure also. Moving to the animal kingdom, they are also having certain features, general features, heterotrophic nature. Heterotrophic nature. So, they are in general heterotrophic in nature, depending on others for their food. Multicellular, they are made up of many cells and these cells lack cell wall surrounding them. They depend on certain other organisms like plants or other animals also for their food and energy. They are having certain internal digestion, they are having certain food storage also, holozoic nutrition, they are engulfing as such the food, they are having definite growth patterns also, and sensory neuromotor mechanisms also for stimulus response. They show a particular locomotion also, and sexual reproduction. Moving to the virus, basically these are the structures which are living when they are in the host, they are non-living when they are outside the host,
yes.
And they are having a protein coat surrounding them and a genetic material which may be a DNA or RNA also, and they utilize host body for metabolism and replication. Similarly, there are certain structures which are called as viroid. So, they are not having that protein coat, they are having only RNA as a genetic material, and they are infectious also and of low molecular weight. And prions, when we discuss about the prions, they are those infectious agents which changes the protein folding, the type of protein folding, they disturb that protein folding. So, these are those prions which are infectious agents, and abnormally they change the folding of the proteins abnormally. And moving to the last part of this session, the symbiotic partnership of the lichens. As already we have discussed the symbiosis, that is symbiotic association. It is a mutual cooperation of two organisms coming together. In the case of lichens, one is phycobiont, that is algae part, and another is the mycobiont, that is the fungi part. They both come together, live together, and this association is called as symbiosis, and in such a case, this organism is called as lichen.
Right.
Thank you.
Right. So, it was really wonderful how Dr. Puneet Sharma told us all about living world and biological classifications. Now, at the end of the session, I would like to give my thanks to Dr. Puneet Sharma for such a patient and detailed explanation of our topic for today. Sir, बहुत-बहुत धन्यवाद आपका हमारे साथ जुड़ने के लिए. हमारे प्यारे दर्शकों, अभी हमने बायोलॉजी का एक सेशन अटेंड किया जिसका टॉपिक था लिविंग वर्ल्ड एंड बायोलॉजिकल क्लासिफिकेशन. इस टॉपिक में हमने बहुत सारी चीज़ें डिटेल में सीखी. बायोलॉजी क्या होती हैं, बहुत सारे माइक्रो ऑर्गेनिजम्स के बारे में जाना, प्रिजर्वेशन टेक्निक्स के बारे में जाना, बायोलॉजिकल नेम्स किस तरह से दिए जाते हैं ऑर्गेनिजम्स को और प्लांट्स को, उसके बारे में भी जाना. दर्शकों, अगर आपने कुछ मिस कर दिया है, सो यू कैन गो बैक एनी टाइम टू आवर यूट्यूब चैनल and catch the content which was telecasted throughout this 1 hour there also. Now, for the upcoming hour, I would like to share we are having a session for class 12 students for physics, and the topic that we are going to take is current electricity. So, viewers, don't go away anywhere. We will be back very soon. Keep learning, keep growing. Namaskar for this session.